like a lot, doesn't it? Well, little worms that have only 5 chromosomes have up to 19 million genes, so it actually isn't that much more than very simple organisms. And of all of those 3.8 billion base pairs, only 2% of them actually encode those 20 million genes. So 98% of our genetic material doesn't necessarily encode anything, but it does something. What it does, we have some ideas, but we don't know for sure. Now, of those 20 million genes, this is essentially what they are. You can see the largest chunk are unidentifiable, about a quarter of our genes. We don't necessarily know what they do. Some of these other ones are transporters and ion channels. A lot of them are involved in the whole cellular machinery, the things that make our cells produce energy, the things that make us use energy and live, all of these little things. Now, that long chain of DNA is so big, it can't necessarily be all kind of left out in one long strand, or we wouldn't have enough room in our cells. So our body's actually created a really cool way of packing this down. You take your double helix, which is at the very top over here on the right, and they bind to these little proteins that they can coil around. They coil around those and they become what we call chromatin, which is a condensed form of DNA. Eventually, they coil back on themselves some more and twist up. It's almost like a braid within a braid within a braid. They all come together to form a chromosome, which is at the very bottom here. And I have a very pretty picture coming up here of all of our chromosomes. This is what we call a karyotype. It's just a specially stained picture of the 23 pairs of chromosomes. And the way they kind of came up with chromosome 1, well, 1 was the biggest, it was the longest. 22 is the shortest down there at the very bottom. X and Y, those are our chromosomes that make us genetically male or female. But essentially, this is it. This is it. This is all the genetic material necessary to create a human being. Now, why do we have pairs, though? Pairs of chromosomes. It's a really interesting kind of concept. Why not just have one for each when we need less genetic material? Well, we have genes on each one of those chromosomes, and they have a complementary gene or an identical gene on another chromosome. Let's say you have a mutation in one of those genes, well, or one of them, that mutation causes something to not necessarily produce the correct gene. Well, you have one on the other side that will. And so it's kind of a backup plan for your cells to have these backup genes, but also the more interesting thing is having different copies of genes can produce different results when we talk about traits. What we say as doctors or geneticists, they say phenotype, what you can see on the outside. So one of the most simple examples is eye color. We know that blue eye color is what we call a recessive trait. So it means that you have to have 2 copies of the recessive gene to actually have blue eyes. So in this little model down in the bottom right-hand corner, you have 2 brown-eyed parents. They have the dominant brown gene, but they also have a copy on their other chromosome of the blue eye gene. You can see that if they had children, that 3 out of 4 would be likely to have brown eyes. It's just an idea about how multiple copies of genes can produce multiple results. But it's not quite simple. And we know this because it's not all black or white. It's not all brown eyes or blue eyes. There's hazel, green, and grayish color eyes, right? We know that hair color can vary, and again, this is just another way that our bodies have something in their genetic code that we don't completely understand yet. Our DNA, yours, mine, it's 99.9% similar. That's pretty remarkable. A lot of us look very different. A lot of us have different illnesses. A lot of us may have different metabolisms. But we are 99.9% the same. And I think that this shows a couple different things. Very small changes in your DNA, very minute changes, can make a big difference in what is actually produced. as far as hair color, eye color, skin color, whatever it might be, your ability to metabolize sugar, develop diabetes, things like that, but also that our environment definitely plays a role into which genes get expressed, which proteins get produced. And I think that I'm going to hopefully demonstrate this a little bit further as we go along, but there are a lot of factors that are at play, which we don't completely understand. So speaking about genetics and disease, the first example I'd like to bring up is a very simple one. For a long time in the study of genetics, we've looked at diseases and tried to understand more about our genetic code by studying diseases with very specific phenotypes. So Huntington's disease is an unfortunate neurodegenerative disorder that affects people. It causes uncontrolled movements, emotional problems, and problems with brain function. It's caused by a single gene that we've identified called the HTT or Huntington's gene. Mutations in this gene lead to development of the disease. Pretty simple and straightforward. This is an interesting gene in that, or the interesting mutation in that most of us have this gene, and we have what we call these CAG, trinucleotide repeats. That is the specific mutation, is that this little sequence of DNA just keeps getting repeated. Now, if you have more than 35 copies of it in your gene, more than 40, you start to develop symptoms of the disease. In subsequent generations, in people that have Huntington's, it tends to occur earlier in life, and we find that's because there's more copies of that. Now, this disease is what we call a dominant mutation or a dominant disease in the fact that you only need one copy of this. Remember how I said you had a backup copy in your other chromosomes? Well, in this one you don't necessarily have that safety because this is an autosomal, what we call dominant inheritance. So by having just this single mutated gene, you're going to develop the disease. Now, a more complicated example of how genetics play into diseases is breast cancer. So we know that there are certain genes that are involved with the development of breast cancer. BRCA1, BRCA2, and P53. These are genes that our bodies have that repair DNA. So let's say you're exposed to radiation. Let's say you're a medical student and you're eating junk food all the time. Your bodies get exposed to chemicals, things that could potentially damage your DNA. By damaging DNA, our bodies have an intrinsic set of machinery that can repair it. Well, when these repair mechanisms aren't there, sometimes cells then will grow uncontrolled. That's what happens in a tumor. It's uncontrolled cell growth. So BRCA1 we know is a DNA repair enzyme. And when it's damaged, it can be a cause, or one of the causes, to develop breast cancer. But just by having a mutation in that does not mean you're going to develop breast cancer. Actually, only about 15% of people with breast cancer have a BRCA1 mutation. And it's not the only gene that we've identified in breast cancer cases. It's multiple genes. We find that there are other factors that influence the development of breast cancer, like your diet, alcohol consumption, being exposed to radiation, decreased activity. These things are risk factors that you can modify. So I think this shows that there are genetic causes here in the case of this disease, but there are other things which you can do, that you have the ability to change, which could reduce your risk of developing that disease. And so at this point, we'll just do a quick little summary. These are some of the more important points that we've kind of touched on thus far. We are 99.9% identical to each other. We have no control over the genetic material we have. We got that from our parents. Just because you have a mutated gene does not mean that you are going to develop that disease. Gene expression depends on a number of factors, which I think is most important to highlight that it includes your environment. And we also have control over those factors, and that can determine how certain genes are expressed. So we're going to move on to the second part of this talk too, which is talking about how genetics can be involved in development of chronic pain. And so there's a lot of research out there now that looks at this. The big things that we're trying to figure out is there's a big difference in how everybody senses pain. Some people say, oh, I have a high pain tolerance, right? And certain people we know that have certain diseases like CRPS can have incredible sensitivity, and stubbing a toe can be exquisitely painful. We know that people respond differently to pain medications, and we're starting to understand more about why that is. And probably one of the most interesting things, I think, is determining who is susceptible to developing chronic pain. If we can get ahead of this problem before it actually occurs or rears its ugly head, I mean, that's the key to it right there. And so far, there's been hundreds of genes of interest identified. And we're going to talk about some of them today and about what some of them actually do. So, in developing chronic pain, we've kind of always looked at this as a transition. Acute pain, a traumatic injury, so a bad soft tissue infection, surgery, a bad accident, whatever it might be. That can develop, or that can cause I don't know how many patients of mine that have heard them tell a horrific story about how their chronic pain started. But the truth is not everybody that has nerve damage or a bad accident necessarily develops chronic pain. So it's kind of interesting, like, who is actually going to be developing it? So we know that because A does not necessarily lead to B in this situation, that there has to be something else kind of going on. We know that that environmental kind of exposure also has some effect on your genetic material and how it's expressed, and that there is a very variable kind of intensity of pain that people feel from a different set of insults, injuries, and the responses to medications can differ. So we know that there's a genetic basis here. We know that genes are at play. And when we talk about identifying genes, I think I mentioned earlier that we have looked historically at genetic diseases in very specific populations, small populations with very, very clear diseases that we're looking for. And so when we talk about chronic pain, the genes that we've identified are ones in very specific populations. And so low-hanging fruit here, these are the genes that have been identified in, say, these hereditary sensory and autonomic neuropathies. There's familiar periodic fevers, and we'll go into some of these, but these are genes that have been identified in syndromes that feature pain as one of the prominent symptoms. So, in these genes, these are the types of genes that they encode. So in familial hemiplegic migraines, we know that it's calcium channels, sodium-potassium channels, and sodium channels which have been involved. We know in primary erythromelalgia, sodium channels are mutated. We know in paroxysmal extreme pain disorder and familial episodic pain syndrome that sodium channels are involved. The sodium channel is ubiquitous in your nerves. Sending a nerve signal along the length of a nerve is dependent on sodium channels opening and allowing an influx of certain sodium and potassium. So these ions that travel in and out of our nerves are the ones that cause the sensation. And so it makes sense that these channels would be involved. We're talking about the disease on that kind of a level, on the molecular level, when we're talking about the genetics of these things. And so, moving on here, what we're seeing, though, is in most people, they don't have these very interesting and rare familial or genetic syndromes, but they do have symptoms that mimic them, or symptoms that are similar in those syndromes. And the developing chronic pain is probably a combination of multiple genes. Maybe you don't have one of those mutations in one of those sodium channels. Maybe you have one good copy, one bad copy. So maybe your body's making some good ones, some bad ones. There may be certain ones, like in the breast cancer syndrome, that there are certain genes that are involved in one type of cell signaling that gets overexpressed. So maybe if you do have gene X, your risk of developing chronic pain is increased by 15%, right? But we know there's a lot of other ones, and we know that smoking, exercise activity, those things can play a role. So we know that there are environmental factors that influence all this as well. So if you look at the trend of discovery of genes involved in pain, and these are just the genes. We don't necessarily know what they encode, but this is the pace. You can see since the beginning of the 21st century, the discovery of genes involved in pain syndromes has gone up dramatically. So this is a real hotbed of research. Some of the genes that we've identified. Carboxyl methyltransferase. It degrades dopamine and epinephrine and norepi. These are your body's chemicals that kind of kick up that fight or flight response, cause your heart to race. If that's not degraded, well, your body is never going to really turn off some of, let me back up. So when that's involved, when you have like your flight or fright response, you tend to ignore painful areas. In fact, some of the medications that are actually used to treat pain, such as Cymbalta, Savella, they actually increase these chemicals, norepinephrine and epinephrine. Those are good chemicals if you're talking about neuropathic pain, because all of a sudden your body shuts down a lot of those nerve signals coming up to your brain, right? Because if you're being chased by a bear in the woods and you sprain your ankle, you're not going to be paying attention to that pain, right? You're going to be running away. You're going to be able to get away. That's the fight or flight response. Now, if this is damaged and you're not degrading these things, or if you have a propensity to degrade more of it, then you're not going to have a lot of these inhibiting a lot of those nerve signals coming up. So that one makes sense. There's the immune system, this HLA, human leukocyte antigen protein, that regulates your immune system. We know that there's a strong interplay between your immune system and certain chronic pain states. Opioid receptors, interleukin-1 receptor. I mean, these are pro-inflammatory receptors here. These are things that can lead to the development of chronic inflammation. A swollen, sensitive, red or purple limb. I don't know if any of you ever heard of that. I have. But there's a lot of these other ones too that are involved with other chemicals that we know are involved in the signaling process for nerves. So what about our responses to pain treatment? I think I mentioned just a moment ago the way that Cymbalta works in Savella and how it works, but there's a whole lot of other chemicals along the way when you take a pain medication. When you take any type of medication, you need transporters to bring it into your cells. Sometimes you need enzymes in your own body to activate it. So you can see the E's and T's and the R's up there. Those are all different areas where there could be potential mutations or what we call polymorphisms, slightly different variations person to person. This is where that 0.1% might be a little bit different for you than me. And so we know some of these exist. We've actually looked at a lot of the cytochrome enzymes. In fact, sometimes I'll actually test patients for these sort of things if they're not responding to certain pain treatments that I've tried. But you can see the opioid ones are very common. There can be different variations in how people will metabolize these medications, and metabolism is necessary for them to actually work. But it's not just opioids. It's anticonvulsants like gabapentin, Lyrica. It's ketamine. It's anti-inflammatories. You can see there's a whole lot of them here that can be a little bit out of whack. And if they are out of whack just a little bit, it may be the difference between a reasonable amount of relief and none. It may be the difference between having an allergic reaction to something and not. But I think what I've tried to do today is introduce this idea that it's a combination of our genetics environment. We know that for sure. There is so much more to our genetic material that we don't understand. That 98% of our DNA that doesn't encode for proteins, it does something. It's there for a reason. Our body wouldn't keep it around if it didn't. We know that there are other things in our cells that we haven't necessarily pinpointed, but there's microsomal RNA, mitochondrial DNA. There's this whole field of epigenetics. Epigenetics looks at the way that your DNA is packaged, the way that it's tagged with little different chemicals called methyl groups. And that has to do something. And just recently, this new genetic tool came out called CRISPR. It is really fascinating, but what it can do is actually it can alter genetic material in living cells. And when I say alter, it can alter specific letters in your genetic code. In fact, last week they just released that in the UK, there's a scientist he's originally from Kazakhstan. He used CRISPR to actually eliminate a single gene that was mutated in these human embryonic cells that caused hypertrophic obstructive cardiomyopathy. It's a cause of sudden death in young folks. Pretty amazing. Now, the same group, or not the same group, but the same tool is being used to see if they can sterilize mosquitoes. If you sterilize mosquitoes and they couldn't reproduce, you'd cure malaria. There'd be no more West Nile virus. Zika virus would be gone. And this is where this stuff gets really exciting. There are certain individuals in China that have a mutation in one of their white blood cell receptors that makes them resistant to getting infected with HIV. What if you could introduce that in the world population? HIV would be irrelevant. We're close. We're closer, I should say, closer to being able to find out if there are specific genes and which ones are the most important ones, which ones are the BRCA1, which ones are the P53, which ones are the important genes involved in developing chronic pain. And I think it won't be too much longer before we'll have a way to actually do something about that and potentially cure these syndromes. We're just, I think, at the tip of the iceberg on this. Thank you.
Are there any questions for Dr. Young?
All right, I saw one hand back there first.
I'm wondering, sir, what are the factors that make you say it is worthwhile testing someone for particular genes? When does that make sense?
Oh, that's a good question. So the question was, when do I think genetic testing is worthwhile for a patient? So when I have patients that I see that are having a hard time, it's usually genetic testing if they have a resistance to certain types of medications, and it's usually opioids. And it can be pre-op if I'm seeing a patient before they're undergoing a major surgery, and sometimes if they've dealt with a problem for years and years and years, it's usually when doses are escalated, when I increase a dose, or they tell me they've had a history of using this, that, or the other, and it's never worked. Or increasing a dose to a point, and I think that's more on an individual doc-to-doc basis, but I'll increase it maybe once or twice before I start to think, maybe something else is at play here. It's usually just a send-off blood test. You don't get the results right away, but it gives you an idea how people metabolize things. But it's usually just as an increasing dose doesn't really provide the increased or improved pain relief that you'd expect. Another question.
Well, first of all, thanks for coming in. Second of all, you mentioned in your presentation the example of people getting nerve damage, and in some cases it transforms into chronic pain, and in other cases it just stays in acute pain. So what if you don't have an acute trauma like that, but let's say you do have a genetic connective tissue disorder? Are there
Okay, so the question was, in certain genetic diseases, say like Ehlers-Danlos, where you actually have a known mutation, is that sufficient? I think you were asking, is that sufficient enough to lead to chronic pain, or are there other genes at play in that situation?
Or what explains, for people that have that, what explains variation in pain experience?
Gotcha. Okay, so the variation in pain experience in patients that have a specific genetic disorder that's known and diagnosed. Okay, so I think that it's interesting, right? Because not everybody with Ehlers-Danlos necessarily suffers from repetitive joint dislocations and chronic pain because of that. Even certain ones sense it much more sensitively than others. And I think that's a role for some more of these studies. And I think that's probably where you're going to find that other 98% of our genetic material does something. I mean, it's all tightly packaged together, but your body still has the ability to get little tiny proteins in there, unwind the DNA, and transcribe what needs to be transcribed. And there probably are certain tags in those individuals, certain chemicals that are linked to those parts of your DNA, perhaps, that will lead to, I guess, more transcription of a factor that maybe makes your nerves a little more easy to set off or more easy to fire. It might be something like that, but I think that probably what you're talking about there is, you're just probably, that's a nice example of what I would call a polygenic type situation where multiple genes are probably at play. Any questions? Yes?
When a person has had a serious trauma to the body, such as being in a hit and run, and they end up with no type of chronic pain afterwards, and then later they fall and they have a slight trauma to the body and they end up with terrible chronic disease afterwards. How does that genetically work out?
Oh, that's interesting. That's a hard one to answer, though. I think at different stages in our life, our body expresses different genes, right? So as you're an infant and you're growing, your body is quite commonly expressing genes that are important for organ and brain development. As we get older, a lot of our genes, certain genes get turned off. We may not make we're not going to, you develop certain organs, and then after that, those genes are not necessary to develop those organs anymore, right? After you're a child and you develop them, there's nothing else to do. They just continue to grow. And so it may be a product of which genes are expressed at which point in time. And if, as we age, certain genes may be turned on that may predispose us. We know that in younger patients, especially younger patients with CRPS, the rates of remission are much higher than middle-aged and older folks that develop it.
Hypothermia. Patients who have malignant hypothermia, and also if they have issues in the methylation cycle where they cannot break down certain chemicals to make them methylated. So therefore, there are some medications that they will not be able to break down physically. How does one know? Because from my experience, going through pain management, they did not do genetic testing, and I dug it out on my own due to my medical background, figuring out how to address certain issues. Is there a test that you guys utilize when you have patients coming in because you don't know? Malignant hypothermia is inherited genetic defect, and it can affect how you metabolize medications. Pain medications may not work appropriately for these people. So as a patient, how can we be proactive with our doctors to try to find the right medications to hone in instead of going through medications that aren't necessary?
Could we put up I had one more slide at my very end of my talk. Could we put that back up? The very end of my presentation. The very last slide. So that's an interesting, it's the use of genetics and genotyping in the treatment of patients currently, I think is still pretty controversial. I do it for pain medications when I say that patients don't necessarily have the response you expect. I think ultimately this might be a little bit of an extreme example, but I think one day we may all be implanted with a chip that has our DNA in it, right? It might have our entire genome encoded on a little chip. I could see that happening. Go back. And what could happen is your doctor could scan that, and maybe they'll have a list of medications that print out and say like, oh, these are the best ones for your body. But we don't have that information just yet. We don't have that ability to do it just yet. And I think that there are no agreed upon genetic tests by doctors. I mean, I think it's still done on such an individual doc-to-doc basis. Some, I know, don't do this at all. I do it sometimes, not commonly.
What about the factor of different patients metabolize medications at a slower rate or a faster rate?
Yeah, and those are the cytochrome polymorphisms that I was pointing out before.
Right, with genetics.
Yeah, I still think that most patients still metabolize things pretty similarly. We do have outliers, definitely, and I think being able to better identify them there, that's important. We haven't really figured out a better way to a screening process to formally figure out who needs it. I don't think necessarily everybody needs to have it done, but people that don't necessarily respond in the way you'd expect are the ones that I usually test.
On a personal level, I do have malignant hypothermia, and I do metabolize things very rapidly. And if patients, I think education as far as in pain management, that patients to become aware of if they have this particular genetic defect, that they become aware of medications that start low.
Thank you. There's a question in the back from before. Somebody had their hand up. All right. We can take one more question.
Do you have anything ongoing on the role of genetic modifications in central sensitization, either experimental or clinical?
The question was about central sensitization and current or ongoing studies involving them. Not to my knowledge. This is a big area of research interest for me personally. I was in high school and I took some genetics classes at a community college. I did 2 years of molecular genetics research after college, and this is something that it's fascinating to me, but it's also something that I have a very strong interest in. And with the new genetic tools that we have available, like CRISPR, I really do think that we're going to see a major change in how we kind of look at disease and, more importantly, how we treat disease, potentially curing it. All right. One more question. Okay.
Okay. I understand there's a test out that can help determine how your body's going to react to different medications. Are you familiar with that test? As far as I understand, insurances aren't covering it, but it helps to determine whether certain medications, cholesterol medication, and I wondered if it was also for pain medication.
Yeah, I am familiar with there actually are several different proprietary, I guess, tests out there right now. And so they're different. I've had, I don't know, 4 or 5 come by my clinic and say, hey, we'd like to offer this to you, your patients. And honestly, it's often a lot of enzyme testing, like we were talking about a little bit earlier, as far as response to treatments. Yeah, it's an out-of-pocket expense, which is no good. What I tend to do is I just pick the few that are of interest to me, and I send them off. And so it tends to be a little bit, and they get drawn in my lab at the hospital. So I don't think my patients get hit with a big bill because of it. But yes, they are an out-of-pocket expense. There are several different kinds of companies out there, and they test these polymorphisms, not just the ones I listed, but many more, looking at the different medications that people will commonly take for diabetes, like cholesterol, high blood pressure, heart disease, those things.
And they help with the trial and error process and help make it more.
Yeah, the idea is that you can streamline that process of trial and error and not even try the ones that your body's not going to respond well to. It's not necessarily going to predict whether you have an allergic reaction to it, but it will predict your response.
And is that applied for pain medication also?
Yes, it does apply to pain medications as well.
And what kind of test, do you know the name of that kind of test?
I don't know the name of anyone specifically. She'll answer your question. She'll come by and introduce herself in a
Okay,
moment.
thank
All right?
you so much.
All right, thanks everybody.
Thank you.
Have a great day.